of enzymes. There is classification of enzyme. First, the name of an enzyme in many cases ends in ASE or ASE. For example, the enzyme for the hydrolysis of sucrose is sucrase. The name describes the function of the enzyme. For example, oxidases catalyze oxidation reaction. So sometimes enzymes are named according to the substrate those are catalyzing, and sometimes the reaction type, for example oxidation reaction. Sometimes common names are used particularly for the digestion enzymes such as pepsin and trypsin, which are protein hydrolyzing enzymes. Some names describe both the substrate and the function. For example, alcohol dehydrogenase, alcohol from the substrate, and dehydrogenase, which causes dehydrogenation reaction. So it oxidizes alcohol. Enzymes are classified according to International Union of Biochemistry and Molecular Biology, which has recommended a system of nomenclature for enzymes, and according to them each enzyme is assigned with two names. One is trivial name or common name or recommended name, and other is the systematic name or official name. The formally used term of the enzyme are ferments, because those were involved in the fermentation process, and that was based on the fact that enzymes are basically causing fermentation. First discovered enzymes are given name according to the source or method of discovery. Their names are therefore without any relation to the reaction mechanism. Many end with in ending. An example is pepsin, discovered in gastric juice, from Greek pepsis mean digestion, or tyalin, found in saliva, from Greek tyalon, which means saliva. And recommended nomenclature for enzyme, simpler than the systematic nomenclature, but yet being systematic in a way. The recommended nomenclature is often used in a routine practice. The name is made by combination of substrate plus ASE. For example, amylase, which catalyzes the hydrolysis of amylose, and a type of reaction plus AS, for example dehydrogenase, causing dehydrogenation. According to the systematic classification, enzymes are classified into 6 functional classes, which are named EC1, EC2, which is also called as EC number classification by the International Union of Biochemistry and Molecular Biology on the basis of types of reaction they are catalyzing. For example, oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. So the biochemical properties of these enzymes include oxidoreductases are involved in many chemical groupings to add or remove hydrogen atoms. Transferases, these transfer functional groups between donor and acceptor molecules. Kinases are specialized transferases that regulate metabolism by transferring phosphate from ATP to other molecules, for example adding phosphate to glucose. Hydrolases, which add water across a bond and hydrolyzing it. Lyases, which add water, ammonia or carbon dioxide across a double bond or remove these elements to produce double bonds. Whenever these molecules are removed, a double bond between the neighboring elements is created. Isomerases, which carry out many kinds of isomerization, for example L form to D form isomerization, which result in mutase reaction, shift of chemical groups, and others. Ligases, which catalyze in reaction in which two chemical groups are joined or ligated with the use of energy from ATP. Each cause food spoilage, rotting, and this autolysis of food is prevented by putting the food in refrigerator or through heat treatment that is called as blanching. Responsible for changes in flavor, color, texture, and nutritional properties. These can be activated by heat to extend storage stability of food, for example, through blanching, and used for fermentation purpose in foods, and can be extracted and purified. These enzymes also find application in the food industry, for example, carbohydrates production, which are used in production of corn syrups from starch, because the starch further yields the small molecule which have more sweet taste. Example is glucomylase. And conversion of cereal starches into fermentable sugar in malting, brewing, distillery, and baking industry, for example, amylases. Similarly, proteases are used for meat tenderization process. Example is bromelain, papain, and lysine. Lipases are there for flavor production in chocolate and cheese. Denaturation of these enzymes occurs at 40 degrees Celsius or higher temperature. Too much heat slows and stops enzyme activity. This destroys enzymes that alter the food quality, for example, blanching vegetables to prevent spoilage during frozen storage. These enzymes are also denatured or affected through change in pH. Changing the pH can speed up or slow down or even stop the enzymatic activity. For example, lemon juice is added to apple, citric acid is added to shrimps, which reduce the enzymatic activity. Sometimes enzymes are also denatured with salts, like the common salt is used in food production, which binds to the enzyme in food that causes spoilage. This salt is also used in canning. It decreases the microbial growth. Water in foods causes bacterial growth, and adding salt to this water reduces the microbial growth or bacterial growth. Salt causes osmotic shock to bacterial cells and even leads to cell death. Sometimes there are enzyme inhibitors are there which inhibit the enzymatic activity. Naturally occurring enzymatic inhibitors include egg whites, pesticides that are naturally part of a plant structure, marigold, etc., and synthetic compound, for example, drugs. These are enzyme inhibitors and can be used for reducing their activity.